to today's Public Health Ontario Rounds presentation on leveraging mucosal immunology to design better vaccines for respiratory viruses. My name is Shelley Bolton, and I'm a scientist in health protection at Public Health Ontario, and I have the pleasure of moderating today's session. Before we begin with today's presentation, I would like to acknowledge and respect the lands, sky, and waters of Ontario, including for their contribution of sharing to support all life within. I acknowledge and respect the treaty, unceded, and traditional territories of all First Nations across these lands and waters. I acknowledge and respect the traditional territories and communities of the Métis Nation of Ontario. I acknowledge and respect Inuit connection and contribution across these lands and waters. Please join me in acknowledgement of respect of all Indigenous peoples, their life journeys, worldviews, and unique indigeneity. May we respect and honor the many connections, relationships, and life journeys of Indigenous peoples and indigeneity to the lands, sky, and water across all Ontario since time immemorial to this very day. May their wisdom always guide our own paths forward with open hands, open hearts, and open minds for the mutual benefit of all in Ontario. To the author of this land acknowledgement, Daniel Coles, thank you very much for sharing the land acknowledgement with us. So before we kick off, I'd like to mention a few housekeeping items. Firstly, the chat pod has been disabled in order to limit any distractions during the presentation. We will have a discussion and question period after the presentation. And if you have any questions, then you can use the Q&A pod that is available at the bottom of your screen. If at any point during the session you experience any technical issues, you can email capacitybuilding@oahpp.ca. I'd like to state that as the moderator of the session, I have no conflicts to declare. So it is now my pleasure to introduce Dr. Matthew Miller. Dr. Miller is the director of the M.G. DeGroote Institute for Infectious Disease Research at McMaster University. He is also an associate professor of biochemistry and biomedical sciences and holds a Canada Research Chair in viral pandemics. Dr. Miller completed his PhD in microbiology and immunology at the University of Western Ontario, where he studied the molecular virology and pathogenesis of DNA viruses. Dr. Miller has been heavily engaged in COVID-19 research, including the development of next-generation vaccines, therapeutics, and diagnostics. He is a member of the National Advisory Committee on Immunization, COVID-19 Working Group. So Matthew, we are excited for your presentation. Over to you.
Thank you very much for that introduction, Dr. Bolton, and it's a real pleasure to have an opportunity to talk to everyone today. Just to get started here, I want to share my disclosures. I've served on the Scientific Advisory Board for Seqirus and Sanofi and as an expert consultant for BC Hydro. I've received funding from several sources, including CIHR, NSERC, CFI, ORF, CITF, the Canada Research Chairs Program, and Providence Therapeutics, and I'm co-founder of Aeromune Biotherapeutics. The objectives of today's session are really to talk about and be able to describe the unique characteristics of mucosal immune response to pathogens, and to understand in particular the rational basis for mucosal vaccine design. In addition, we'll discuss ongoing developments in both the preclinical and clinical evaluation of mucosal vaccines for respiratory viruses. So although we're still reeling in the midst of the COVID-19 pandemic, of course, we're no stranger to pandemics. Over the course of the last roughly 100 years, there have been 5 influenza virus pandemics that preceded our current COVID-19 pandemic. The most serious of which was the 1918 H1N1 Spanish flu pandemic, and the most recent of which was the 2009 H1N1 swine flu pandemic. One of the key points that I'm going to come back to over the course of today's talk is about how we poise ourselves to mitigate the impact of pandemics. And what I'm showing here is the global number of confirmed cases and deaths. This is a little bit out of date, but as of the spring of this year. And what I'm going to show you in the hatched line now is when COVID-19 pandemics were, or COVID-19 vaccines, I'm sorry, were first approved in Canada. And as I'm sure this audience knows, this is really, I think, one of the tremendous medical miracles of our time. In ostensibly 1 year, we were able to both develop and approve extremely effective vaccines against a family of viruses for which there were no existing vaccines on the market. Now, of course, the effectiveness of these vaccines have been challenged, especially more recently. And this is due in large part to the evolution of COVID-19 variants of concern, who have antigenic properties that lead our immune response to be less effective against the variant than they were against the vaccine antigen, which was derived from the ancestral or Wuhan-like strain of the virus that started this pandemic. Now, many people ask me, could anything ever be worse than what we've experienced over now going on the last 3 years? And what I tell them is indeed it could be much, much worse. If we look back to the 1918 Spanish flu as an example, this pandemic was widely considered to be the greatest natural disaster in human history. And this is largely because of the tremendous morbidity and mortality toll that this pandemic exerted over a very, very short period of time. Of course, as humankind, we've suffered the scourge of many really problematic infectious diseases in our history, things like plague, HIV, smallpox, but these infectious diseases circulated over much larger time frames. In 1918, over the course of roughly 2 years, between about 1918 and 1920, this Spanish flu resulted in the death of 3 to 5% of the global population. And if we extrapolated the population in 1918 to today's population, that would amount to roughly 370 million people, which is about the combined population of Canada and the US. By contrast, with the COVID-19 pandemic, we're going on 3 years, and there's been getting close to 7 million deaths. So much, much lower than what we would have experienced were this Spanish flu to have occurred today. There have been 3, arguably 4 influenza pandemics since 1918, and none have been as severe, but this isn't by any, miraculous human intervention. This is essentially by sheer luck. And prior to the arrival of the COVID-19 pandemic, what I always used to say is that what really worried me is that despite the tremendous medical advances that have happened over the course of the past 100 years or so, we'd really made no major improvements in pandemic preparedness. And that's for a few reasons. One is that, we lack vaccines and prophylactic measures to mitigate the impact of pandemics before they emerge. And secondly, we have very, very few interventions to treat viruses that come from zoonotic sources, both of which really leave us extremely vulnerable as we've experienced over the last 3 years. Now, in 2009, when the swine flu pandemic happened, we were in a very different readiness situation than we were for COVID-19. And that is, of course, because we've been making influenza vaccines, seasonal influenza vaccines, for decades prior to 2009. And there was a huge global infrastructure to both produce and distribute these vaccines. Nevertheless, what I'm showing you on this side in blue is the number of cases of influenza-like illness during the initial wave of the 2009 pandemic. In red, what you can see is the number of monovalent pandemic vaccines shipped, and in green, the number of doses actually administered. And what I hope you can appreciate from this slide is that even in a situation where we know how to make it a vaccine against a given pathogen, and we have the infrastructure in place to produce and distribute that vaccine, we still can't react quickly enough to mitigate the effects of a pandemic, which spreads very, very rapidly. And so we need to rethink our paradigm for how we ready the global population to deal with emerging pathogens. Since COVID-19, there's been a lot of focus by funding agencies and governments and not-for-profit funders like the Gates Foundation as an example, on increasing our ability to respond quickly. There's been the so-called 100 days goal, basically the ability to have vaccine platforms and infrastructure ready to produce a vaccine against a novel pathogen within 100 days. And while those goals are important, I think that it's come at the consequence of overshadowing where the majority of our efforts should really be focused, which is in prevention of these public health emergencies from the get-go. Of course, response is what happens when prevention fails. And, $1 invested in prevention can save us $10 or $100 that we have to spend when we're responding. Not to mention the consequence on human health that results when we're in a response situation. So something my lab has been working on really since the 2009 swine flu pandemic is designing better broadly protective vaccines that would be capable of protecting us in advance of these zoonoses from happening. And what the focus of my talk today is going to be on is really thinking inherently differently about broadly protective vaccines than I think we have historically. Historically, the fields like influenza and HIV have really been focused on antigen selection, which is, of course, important if you want to make a broadly protective vaccine. People look essentially to target conserved antigens in these pathogens, and ideally, not only conserved antigens, but antigens that are less tolerant to mutations, such that if our immune system does target them, they can't quickly make mutations that then render our immune response less effective. However, what we've thought much less about is how the mode of administration of a vaccine affects our response. And what I hope to convince you of today is that this is an equally important consideration. Now, the way in which we're exposed to a pathogen or an antigen or a vaccine has a really profound effect on the quality of our immune response. When we're exposed to antigens systemically, as happens when we get a typical intramuscular injected vaccine, what results is an immune response that's typified by usually strong IgG antibody titers in the circulation, circulating T cells and plasmablasts. In contrast, when we're exposed to pathogen mucosally, we mount a fundamentally different type of an immune response. These responses tend to be characterized by the production of IgA, which is not only the most abundant antibody at our mucosal surfaces, but also the most abundant antibody in our entire body, despite the fact that IgGs are most abundant in serum. Likewise, when we're exposed to antigen mucosally, we also draw in tissue-resident T cells and B cells, and we can also train innate immune cells and mount what are called trained innate immune responses. And these all have benefits when it comes to the ability of our immune system to protect us from subsequent challenge with mucosal pathogens. Now, our propensity to vaccinate parenterally or by injection is, can really be traced back to the early days of smallpox prevention and Jennerian vaccines, where what was done was scab tissue from either smallpox survivors, or in certain cases later from monkeypox, were scraped into the skin of a naive recipient in order to help that recipient mount a protective immune response. And while this is a very convenient way of administering vaccines, and probably a very rational way of administering vaccines that protect us from pathogens that cause systemic infections, it's probably not a particularly rational way to develop vaccines that are designed to protect us from pathogens where the infection occurs via mucosal routes. Now, many, many years before Jenner, in fact, centuries before Jenner, cultures in Asia were vaccinating by a different process called intranasal insufflation. This process was similar to scarification, whereby usually scab tissue from smallpox survivors were ground up, but instead of being scraped into the skin, that material was blown into the nose of a naive recipient in order to generate a mucosal immune response. So I'm going to move on to the next slide now, and introduce to you some work that we've been doing to address this gap in mucosal vaccines, which is the development of a respiratory mucosal next generation vaccine against COVID-19. This vaccine indeed leverages a lot of the concepts I introduced earlier, the targeting of conserved antigens that are relatively intolerant to mutation, while also leveraging a unique mechanism of delivery, which is actually aerosolization, as you can see in the image on the left of your screen. This work was done in close collaboration with my colleague So Zing at McMaster University, and was led by Mike D'Agostino, a very talented graduate student in my laboratory, and Sam Kami, a postdoctoral fellow in Dr. Zing's laboratory. This vaccine is built on an adenovirus backbone as the delivery vehicle. And actually, this really comes from a long history in development of adenoviruses as a platform for vaccine delivery, as well as gene delivery that were pioneered at McMaster University, in particular by doctors Frank Graham and Jack Galdi, starting over 30 years ago. It also builds off of well over a decade of incredibly innovative work by my colleague, Dr. Zing, and Dr. Fiona Smale, who have been working to develop adenovirus-based mucosal vaccines to target tuberculosis. What Dr. Zing and Dr. Smale have shown really elegantly in clinical trials in humans is that when they deliver one of these adenovirus-based vaccines for tuberculosis, either by injection or by aerosolization in human clinical trial participants, the aerosolized vaccine generates both respiratory mucosal immunity as well as systemic immunity, whereas the vaccine, when delivered intramuscularly, is limited to stimulating only systemic immunity. So already we see some of the benefits of mucosal vaccine administration in that we kind of get the best of both worlds. And I'm going to show you that bear out in some of the data that we generated for these new next generation COVID-19 vaccines. In addition, what I want to impress upon the audience, because I think this is non-obvious, is that not all mucosal vaccine delivery systems are created equal. Many of you have probably heard that there's been a lot of work internationally trying to use nasal sprays as a way of stimulating mucosal immune responses. Indeed, flu mist, the seasonal influenza vaccine, is delivered by nasal spray. One of the major limitations, though, of nasal sprays is those spray devices generate relatively large particles that deposit themselves almost exclusively in the nasopharynx, or the upper respiratory tract, basically the nose and throat. And the architecture of the nose and throat and the types of immunity that are able to be developed there are fundamentally different than the immunity we see in the lung. When vaccines are delivered by aerosolization, as our vaccine is, not only do you get delivery in the upper respiratory tract, but you also get delivery of those very small aerosolized particles deep in the lung. And this is really important, of course, because when we get severely ill from respiratory pathogens like influenza virus or like SARS-CoV-2, it's typically because we get lower respiratory tract infections, and that's what leads to severe outcomes. Indeed, what you can see here is, again, in a head-to-head comparison in animal models, if the vaccine is limited to the upper respiratory tract or administered intranasally, as we show in the white bars, you get much weaker immune responses than when that same vaccine is delivered intratracheally, which gets the vaccine deep into the lungs. The challenges associated with intranasal vaccines have also really borne out in COVID-19 clinical trials. Indeed, just over the course of the past month, there was news release of a failed Oxford AstraZeneca nasally administered vaccine trial. Essentially, what the Oxford group and AstraZeneca tried to do was to take their adenovirus vectored vaccine that was delivered via injection and instead deliver it by a nasal spray. And the problem that they had was that when they delivered it by nasal spray, it developed it resulted in the generation of very weak immune responses. And so that delivery platform has now been abandoned. So our next generation vaccine is fundamentally different from all of the currently approved SARS-CoV-2 vaccines in that, in addition to targeting spike, we also target two other parts of the virus, namely the nucleocapsid antigen and the RNA-dependent RNA polymerase. The reason we target these are because we know that these are really important targets of the T cell response, and they're both highly conserved and preserved amongst diverse members of the coronavirus family, including coronaviruses that still currently only exist in bats, for example, but are at risk of causing future zoonotic outbreaks or pandemics. So again, the motivation here is to really develop a vaccine that can maintain effectiveness in the face of a virus that's constantly mutating, and ideally provide prophylactic protection against viruses of the same family that are at risk of causing zoonotic events in the future. The first thing we did was to compare intramuscular versus mucosal delivery of our vaccine. And I think the result that was really surprising to us is that when we delivered our vaccine intranasally, not only did we stimulate excellent mucosal immune responses, but the mucosal administration actually resulted in stronger systemic immunity. As you can see in the graph on the bottom left of your screen, the vaccine delivered intranasally elicited much stronger spike and anti-RBD antibody titers than the exact same vaccine delivered intramuscularly. Of course, these binding antibodies also correlated with much stronger neutralizing antibody responses, as you can see in the middle panel. And as I alluded to from studies done on tuberculosis, only the vaccine, when delivered mucosally, does the vaccine generate lung-specific B cell responses. If the vaccine is given intramuscularly, we don't see these tissue-resident or lung-resident B cells. Now, immunogenicity data is great, but of course, what we really want to know is does this difference in immunogenicity actually matter when it comes to protection against infection? And so we infected these animals with a lethal dose of the ancestral or Wuhan-like strain of SARS-CoV-2. And what you can see in the graph on the left is that the intranasal vaccine protected these animals completely from infection. They don't lose any weight, they don't get sick at all. However, when the exact same dose of the exact same vaccine was administered intramuscularly, you can see these animals get really, really ill. In fact, they look by weight loss initially almost like unvaccinated animals. And almost all of those animals succumb to challenge. Likewise, if we look at viral burden in the lung a couple of days after infection, you can see that the animals that received the vaccine via the intramuscular route do a very poor job controlling virus. They have viral titers that look just like an unvaccinated animal, whereas the animals that got the mucosal vaccine have lung titers that are reduced by many orders of magnitude or many logs. Now, challenging with a matched ancestral virus is, of course, great. But what our vaccine is really designed to do is to be able to generate broad protection. And so the next thing we did was to measure whether or not our vaccine could also protect against immune-evasive variants of concern. At the time, beta was the most immune-evasive of the SARS-CoV-2 variants. And so we challenged these mice with the beta variant. And again, what we found was that the vaccinated animals were completely protected from challenge. They experienced no morbidity. And really importantly, we saw that the vaccine was capable of providing sterilizing immunity in these animals. We weren't able to detect any virus in the lung or brain. For these experiments, we used this mouse model of SARS-CoV-2 called the K18 mouse model. And these mice are very susceptible to neuroinvasion by SARS-CoV-2 because they express ACE2 a little bit unnaturally. It's driven by a non-endogenous promoter. And that's why we're measuring virus here in the brain. This isn't something that you would typically see in humans. Since that time, of course, we want to make sure that our vaccine continues to stand up against the most recent variants, and we've gone on to test whether it can protect against Omicron. And indeed, we also saw perfect protection against Omicron and sterilizing immunity in the lung. Now, what's really interesting about this mucosal vaccine delivery system is that in addition to generating the typical types of vaccine responses that we come to expect with vaccines, the adaptive immunity that's typified by antigen-specific T cells and B cells, this vaccine also very strongly induces a trained innate immune phenotype. Trained innate immunity is very much like adaptive immunity, but it's a property of innate immune cells. And what's really interesting about this is that it's antigen agnostic, meaning that the innate immune cells behave in a way that resembles adaptive immunity, in that when they're re-exposed to pathogen a second time, they respond more quickly and more robustly. However, they can do this to totally heterologous antigens. What you can see in these TSNE plots here, which are a way of depicting complex flow cytometry data in 2 dimensions, and I think this is most clearly shown in panel D on the lower right of your screen, is that when we deliver our vaccines intranasally, we get this characteristic trained innate immune phenotype of both alveolar macrophages and interstitial macrophages in the lung, which is completely absent when we deliver the same vaccine via the intramuscular route. Now, we also wanted to go on to understand what aspects of the immune response were responsible for mediating the protection that our vaccine was able to achieve. And so we did both T cell and B cell depletion assays. And what you can see here is that if we deplete T cells or we deplete B cells, the animals still do fairly well when it comes to their ability to survive a challenge, but they have much higher viral lung titers than what we see in animals that are completely immunologically intact. Also the control animals in the lower right panel here. So we need both T cells and B cells to have optimal control of viral lung titers. And this might have really important consequences as it pertains to the ability ability of the vaccine to protect against things like transmission, for example, which we know is something that's quite difficult for our current vaccines to achieve. We also wanted to know whether all of the antigens in our multivalent vector were actually important for mediating protection. So you can see when we vaccinate with the trivalent vector in blue, we get excellent control of viral lung titers. However, if we vaccinate with a bivalent vector that only contains the T cell antigens, the nucleocapsid, as well as the RDRP, all of a sudden viral titers start to increase. We get less effective control of viral replication in the lung. Likewise, if we give a vaccine that only contains the spike antigen, which is the monovalent vaccine shown in purple here, we also get less effective control of viral replication, indicating that all of these antigens are important and work together to generate this optimal protection that we observe in vivo. Now, one of the things I didn't talk about is how antibody isotype plays a critical role in determining the way that antibodies protect us from pathogens. And as I mentioned in the early portion of the talk, IgA is the antibody that really dominates at mucosal surfaces. And we've been very interested in IgA for a long time because, of course, we're a lab that studies respiratory pathogens like flu and coronavirus. And we actually know shockingly little about how IgA protects us from these infections because it's much more difficult to study than IgG. Now, many of us, when we think of the way antibodies work, I think we have a very simplistic view. We think of an antibody binding to a pathogen, and in the case of viruses, neutralizing that virus by preventing it from being able to infect a target cell in the host. But there are other ways antibodies function as well, and this is by engagement with what are called Fc receptors. So antibodies can sort of be split, roughly speaking, into two domains, the FAB portion, or the FAB2, which is responsible for binding antigen, and then the FC region. And it's the FC region of an antibody that gives it its isotype. So the FC region makes it an IgG1 versus an IgG2 versus an IgM versus IgA. And all isotypes have Fc receptors that are receptors expressed on innate immune cells and some adaptive immune cells capable of recognizing those antibodies. So IgGs are recognized by Fc gamma receptors, whereas IgAs, for example, are recognized by Fc alpha receptors. And when these receptors are engaged by antibody, they trigger the cell that's expressing them to undergo different types of effector functions, which are antimicrobial or antiviral in nature. Now, neutrophils are one particularly interesting cell type because they're the most abundant white blood cell or leukocyte in our bodies, and they tend to express a lot of Fc receptors. Now, neutrophils, when they undergo effector functions, can do things like degranulate and kill cells that way. They can phagocytose pathogens. They're able to undergo antibody-dependent cellular phagocytosis, or basically opsonization of pathogen via ADCP. And they can also undergo ADCC, or antibody-dependent cytotoxicity. More recently, neutrophils have also been described to undergo this really interesting process called mitosis, where they essentially undergo a programmed cell death pathway that leads them to extrude chromatin that can trap and kill bacteria. Now, we wanted to understand how neutrophils respond to influenza virus, and in particular, how they respond to influenza virus when it's bound by either IgG or IgA antibodies. So Caitlin Malarkey, who is now my wife, but at the time was a postdoctoral fellow in Peter Palazzi's lab in New York, did a really interesting experiment whereby she generated immune complexes by mixing influenza virus together with either IgG or IgA antibodies. And what she found was that when neutrophils saw virus bound by IgG antibodies, they underwent a process called respiratory burst, where they generate reactive oxygen species that can help to kill the pathogen that they're responding to. When Kate added cytochalasin D to these cells, which basically prevents their ability to phagocytose the pathogen, this effect went away. However, when she did the exact same experiment, except for this time with virus that was coated in IgA antibodies, she saw something very different, which was that even if she inhibited the ability of the neutrophil to engulf this immune complex, there was still the production of reactive oxygen. So to understand what was going on, Hannah Stacy, an incredibly talented PhD student in my lab who is just about to defend, wanted to know what was happening with IgA. So the first thing she did was to expose neutrophils to increasing concentrations of SARS-CoV-2 pseudovirus. And what you can see is if you expose neutrophils to a lot of virus, you can make them nitose all by themselves. However, what Hannah wanted to know is what would happen if you give neutrophils an amount of virus that doesn't lead them to nitose. So what she did was she took that amount of virus, which is incapable of stimulating nitosis alone, and mixed the virus with either antigen-specific IgG or IgA. And what she found was that, really interestingly, only when the virus was mixed with IgA did she observe these neutrophils undergoing nitosis. Now, this nitosis was dependent on the ability of the neutrophils to recognize this IgA-coated virus complex via CD89 or its Fc alpha receptor. Because if we blocked the ability of that receptor to recognize the immune complex, this nitosis went away. And very interestingly, Hannah also asked, how do different types of IgA do this? So in circulation, IgA is predominantly monomeric, but at our mucosal surfaces, it's in a secretory form, which is a dimer wrapped with this secretory chain. And what she found was that monomeric IgA from serum stimulated potent nitosis, but the IgA at our respiratory surfaces does not stimulate nitosis. And that's because the secretory component on IgA blocks the ability of the Fc alpha receptor to recognize the Fc portion of secretory IgA as efficiently. So what this tells us is that IgA can do different things depending on where it's found and what form it's in. Now, very interestingly, this ability of IgA immune complexes to stimulate nitosis wasn't a flu or even a SARS-CoV-2 specific phenomenon. We could see this with HIV, another important virus that infects us often at mucosal surfaces, and even with citrullinated fibrinogen, which is an autoantigen that's a very common autoantigen in rheumatoid arthritis. And what was, I think, most interesting about this study is that Hannah was able to show that this nitosis actually had an antiviral effect. So when she exposed these neutrophils that had undergone nitosis to virus, those nets were able to literally trap the virus and inactivate it by virtue of the antimicrobial effectors that are studded on these nets. Now, while I've talked a lot about active vaccines, the sort of conventional way we think about vaccines, passive vaccines or monoclonal antibodies are also an important part of our toolbox to protect against pathogens. However, there are many challenges to the effective use of passive vaccines or monoclonal antibodies. This includes that single monoclonal antibodies are very susceptible to having their effectiveness either reduced or diminished altogether if the pathogen mutates. Likewise, almost all of these monoclonal antibodies have to be delivered by IV infusion, which is a very burdensome way, as you can imagine, to deliver a drug. And the real problem in the context of respiratory pathogens like influenza virus and SARS-CoV-2 is that for these interventions to be effective, they have to be given very, very early, usually before someone is sick enough to need to be admitted to hospital. And so how do you identify those people and then infuse them effectively? Well, Ali Zhang, an MD PhD student in my lab, wanted to think about ways of generating a toolbox for broadly neutralizing antibodies that might be capable of protecting us were a new influenza virus to emerge and cause a pandemic risk. So what Ali did was he thought about the way that these broadly neutralizing antibodies that target flu work. These antibodies are fundamentally different than the antibodies that seasonal vaccines are designed to elicit in that they tend to bind this conserved stalk domain of hemagglutinin, which is the viral protein found on the outside of the virus that it uses to get in cells. And what's really interesting is that these antibodies don't do a very good job of preventing virus from infecting cells, but once those cells are infected and they express viral protein on their surface, these antibodies bind the stalk, they recruit new immune effector cells, which then recognize the antibody via an Fc receptor, and the immune effector cell then eliminates that infected cell, much like a T cell would. Now, what's kind of cool is that over the years, we were able to discern that the way that this happens in the context of flu is very unique because hemagglutinin binds sialic acid, which is a sugar that's sort of ubiquitously found on human cells. And while these stalk antibodies were binding flu, hemagglutinin was making reciprocal contacts with sialic acid on the host cell, which make that interaction between the immune effector cell and the infected cell very tight. And it actually enhances the ability of the effector cell to eliminate the infected cell. The problem is that influenza also expresses a protein called neuraminidase. And what neuraminidase does is to cleave the interaction between sialic acid and hemagglutinin. So Ali thought, maybe if I block the ability of neuraminidase to do this, we could make these monoclonal antibodies work a lot better by stabilizing those 2 points of contact that I just talked about. So what Ali did first was an experiment where he infected cells with flu, added antibody, and then added increasing concentrations of oseltamivir, which is a neuraminidase inhibitor. And what he found was that when he added higher amounts of oseltamivir, he could enhance the ability of the immune effector cells to undergo ADCC, their antimicrobial effector function. And this was completely dependent on the ability of the immune effector cell to recognize the Fc region of the antibody. Because in panel E on the lower right of your screen, what you'll see is that we used an antibody with this mutation called D265A. This mutation is just a single point mutation that abrogates the ability of the Fc receptor to recognize the Fc region of the antibody, but has no impact on the ability of the antibody to bind to hemagglutinin, its cognate antigen. If the antibody can still bind hemagglutinin but can't be recognized by the effector cell, adding oseltamivir does nothing, essentially. Now, that's all great in vitro, but what Ali really wanted to know is, would this work in vivo? And so he explored this using 2 models. One model where the antibody is administered prophylactically before infection. This is really, in my opinion, the ideal way to deploy monoclonal antibodies in high-risk people because they're very, very effective at preventing infection. What's problematic is that they have a difficult time protecting people who are already infected because the window to mediate that protection is so narrow. Because for pathogens like flu and coronavirus, it's not usually the virus itself that makes people really sick, rather it's their immune response to the virus. Nevertheless, Ali tested this combination in both a therapeutic and a prophylactic setting. In the top, you can see that indeed, when he combined 6F12, the antibody, with oseltamivir, he got vastly superior protection than either monotherapy alone. And this was also true when he used a treatment protocol. Now, to end off, I'm going to tell you what I think is a really cool story that sort of links together the influenza and SARS-CoV-2 portions of this talk. And this study was led by Andrew Chen, who was a fourth-year student in my laboratory, and was done in very close collaboration with my colleague, Mark Loeb, who's just an incredible infectious disease clinician and an epidemiologist, and who I'm sure many of you on today's rounds know very well. So Mark, over the years, has done a series of really important clinical trials in Hutterite communities in the Canadian prairies to evaluate the indirect protection offered by influenza vaccines. And in the first of these studies, what Mark did was randomize entire Hutterite communities, which are usually about 200 or so people in population, to either getting seasonal flu vaccine or to getting a hepatitis vaccine as a control. And what Mark found in the original studies was that the communities where only the children received the seasonal flu vaccine had disproportionate protection against flu, even in unvaccinated adults in those communities. Whereas, of course, that wasn't the case in the communities that were randomized to get the Hep A vaccine. This was really the first randomized control trial level demonstration of the herd immunity effect in the context of influenza vaccines, and was an incredibly important contribution to how we understand the way that vaccines work, and indeed to demonstrating what important drivers of flu transmission children are. What we did, though, was leverage this study because Mark also systematically swabbed anyone in this study who presented with respiratory symptoms and ran a panel of PCR on essentially all respiratory or all common respiratory pathogens to understand what these people were infected with. And what we wanted to know is it possible that seasonal flu vaccination has any indirect effect protecting against other respiratory pathogens? And what we found, really surprisingly, was that influenza vaccination actually profoundly reduced the circulation of coronavirus in the flu-vaccinated colonies, but not in the hepatitis A vaccinated colonies. Now, of course, the study wasn't powered to initially address this question. It was a post-hoc analysis. So you can see, although our risk reduction is about 50%, you can see the p-value is 0.11 in the total cohort. But if you look to the 30 to 39 age band, where indeed most of our infections occur, you can see that this protective effect was actually significant. Now, the final thing we wanted to do was ask about biological plausibility. How is this happening? And so what we did was to ask, well, is it possible that flu vaccination mounts some sort of adaptive immune response against seasonal coronaviruses? And what we found was, indeed, it did. Most of the infections in this trial were caused by the OC43 beta seasonal human coronavirus. And you can see that in the panel A, people who were vaccinated with seasonal flu vaccine actually mounted strong responses against the seasonal coronavirus. However, hepatitis A vaccinated individuals did not. We don't think that this is true cross-reactivity for reasons I don't have time to get into today, but rather that the flu vaccine boosts underlying immunity to seasonal coronaviruses in previously exposed children in sort of a non-specific or bystander way. With that, I'm up for time, so I'm going to wrap up. What I hope I've convinced you of today is that prevention really needs to be the major focus of our strategy for avoiding the devastation caused by emerging infectious diseases, and that vaccines that offer broad protection against families of viruses that we know are capable of causing pandemics are one really good way to enhance our readiness. Our expectations for these vaccines, though, I think need to be realistic. I don't think it's realistic to think that these broad vaccines are going to be a panacea that achieve long-term sterilizing immunity. What we want is for these vaccines to be able to mitigate severe illness were a new pandemic to emerge. And I think that's a very achievable goal. So with that, I want to thank my lab, most especially, who did all of this incredible work I had the privilege of presenting to you today, and a long list of really excellent collaborators, in particular my colleagues Zoe Zing, Brian Lichty, and Fiona Smale, who are instrumental in the COVID-19 vaccine project, as well as many other colleagues noted on this slide. So thank you very much. And it'll be my pleasure now to pass the floor back to Dr. Bulletin to mediate the question period.
Thank you so much for really an absolutely fascinating talk. We have some questions in the chat. We have approximately 7 minutes, so why don't we kick off? The first question relates to the really interesting mouse model work that you showed for the COVID-19 adeno vaccine candidate. Are those IM-IN experiments done as a primary series on the mice, or were they boosting following an intramuscular prime?
Great question. So in the mouse model, this was a primary vaccine. So it was a single dose. So all of the data I showed you was a single dose vaccine in the mouse model. This vaccine is now in phase 1 clinical trials here at McMaster. But of course, now we're using this vaccine to boost individuals who have received prior doses of COVID-19 mRNA vaccines, because of course that will be the outlook going forward.
So that brings me to another question, and that is, so with some of these mucosal vaccines, they're indicated to be used as a priming dose. Like one example is rotavirus, while for others, including a lot of the COVID-19 mucosal vaccine candidates, are really meant to be a boost. Can you talk about the difference between the two and why we might prefer some vaccines to be primary and others to be used as a booster?
Yeah, it's a really great question. So there's this concept in mucosal immunology called prime pull vaccine, the prime pull vaccine approach. And it's illustrated in many ways, I think, in what we've seen in terms of the really strong immune responses we get in so-called COVID hybrid immunity. So if you're able to generate systemic immunity and then you, via, for example, an intramuscular injection, and then you're exposed to pathogen mucosally, what that does is, by virtue of chemotaxis, it draws in those peripheral immune cells that were generated by the systemic immunization into the lung. Now, in some cases, you don't need to do that, because if you have a vaccine that's capable as a single dose or as a primary series of generating robust mucosal immunity, you might not need to rely on the added benefit of drawing in these systemic responses. But in situations where you have, which is typically the case, by the way, where you have an attenuated vaccine that might not be as strong as a primary series, you can really enhance the effectiveness of that vaccine via this prime pull approach.
Okay, thank you for that. Can you tell us, what do you think is now the major obstacle? And I'm going to say you can also talk about obstacles if there's more than one, in implementing and approving mucosal vaccines for COVID-19 in Canada?
Well, I think one of the major obstacles is actually the lack of immune correlates of protection. So for flu vaccines, for example, seasonal flu vaccines, we use immunobridging studies to assess vaccine effectiveness. And this is really convenient because it means that you don't have to go through phase 3 efficacy trials all the time. We're basically able to measure the amount of antibody in blood and say something about, whether that person has a level of antibody that's likely to be protective. Now, we know that neutralizing antibodies are a reasonable surrogate of protection against COVID-19, but that's all based on systemic circulating neutralizing antibodies. And it's very likely that the correlate of protection for mucosal vaccines will be different. As I told you, systemic immunity and mucosal immunity are fundamentally different in both quality and quantity. And so I think having mucosal correlates will really help facilitate this vaccine design and approval, because right now, what we really want to know is at the stage of doing early phase clinical trials, like phase 1 and 2 trials, what we'd like to know is, can we measure an immune response that we know is likely to be protective to give us confidence that as we move to later stage trials, that the vaccine is likely to work. And barring those correlates, it will be more difficult, I think, to develop these vaccines, or certainly more expensive, because we're more blind going into those efficacy trials than we are where strong correlates have already been validated.
Thank you for that answer. I think we have time for perhaps one more question. Do the T or B cell depleted mice show any other poor outcomes? The survival data looks like they're able to overcome challenge, but are there any signs of weight loss? Viral loads in the lung were high, so just wondering how that might impact disease outcomes.
Great question. This is actually described in more detail in the paper, but again, I had to trim down the story for the sake of time. If you look at the lungs of those T and B cell depleted animals, what you see is that although they didn't lose weight and they survived infection, again, we did see poor virus control, and this poor viral control also resulted in much more lung pathology. So you can see just by gross examination that these lungs have sort of more hemorrhaging in the lobes that are infected, and later on look more fibrotic. And so there does seem to be a very clear role for these cells in helping mitigate some of the downstream effects of healing from that infection, even if someone survives, right? Or even if the animal survives. And I think this is really important, right? Because in vaccinology, especially in preclinical models, we often use pretty severe endpoints, right? Like there's a lot in between life and death. And ideally, we'd like vaccines that not only help people live, but ensure that they don't experience long-term consequences of infection. And again, something I didn't get a lot of time to expand on, but we'd also like vaccines that are capable of more effectively mitigating transmission, because that would really help bolster the public health benefits of those vaccines by reducing community level circulation of pathogens.
I think that's a great statement to end on. So we need to wrap up today. Thank you so much to Matthew for presenting. Thank you very much to everyone who took the time to join us. You can expect to receive a brief and anonymous PHO round survey for today's session, and we encourage you to fill it in because it really helps us with our programming. Lastly, to access past PHO round presentations and view confirmed upcoming sessions, please visit the PHO website, head to Education and Events, and then click on Presentations. So with that, we are signing off. Thank you and have a wonderful day.